So as for the COVID-19, so here I'm gonna introduce some of our in-house experience at GIDI when the outbreak first happened, and as for a group of mostly computer scientists and chemists, and with very few virologists involved, that we would like to figure out a way to to search for the therapeutics for this completely unknown and new pathogen. So this here is what we did. So first is we query these questions to a system that full of data-driven algorithm and pile of data and literatures. And then first we perform data mining and cleaning to find relevant evidence, and then we reason on those evidence to extract the most important piece of information and use those information to build AI models, and then use that model to screening new possibilities for the treatment. So this is a high level, and then the following talk, I'm going to give a stepwise introduction to each of this workflow. So first of all, let's talk about why this workflow could work. So first, let me talk about some advantage. First, each steps have been achieved by a computer program in this workflow. So that means, and they can be integrated through API if they haven't been integrated yet. So which means even individual tasks may be done separately on a web base or a separate computer program. But as long as it is a computer program that does the job, it can always integrate together. The second one is it requires online resources and global collaborations to achieve this task, because different steps require different servers and data from different countries and different research groups. And the third, the entire workflow, if completely automated without any human adjustment, the only cost like less than a day to finish all the computing and analyzing job on a typical high performance computing system. And then the last one is the expert intervention is minimum. So basically, we would like to build an AI system that could be as insightful as a experienced virologist in this topic, so that it can respond very quickly in everywhere in different research community to bring everybody up to speed. But however, the current AI technology in drug discovery is still under development. So the workflow is not completely automated right now and requires some human AI augmentation, that's the word I like to use, so that to help bridging the gaps between different type of programs. So first of all, I just wanted to highlight one astonishing event that the global collaboration has been going on GitHub on the COVID-19 combat battle. That this screenshot has been identified today. That there is 31,000 over repository that has been created on GitHub across the world, and I remember this number was 3,000 at early February. So this is very impressive, and I will introduce some of the project that is one of the 31,000 project later. So first, let's talk about the first step is when we get a new virus, we need its gene sequence. That's the first, at least the minimum amount of data we would need to figure out the approach, so that we get that gene sequence of this new virus on January the 5th on GenBank. And that's the first deposit of this unknown virus. And then we do a quick BLAST. BLAST is a searching algorithm that aligns different local regions of a known genome against the entire genome repository to find the most similar ones. And it has been used extensively in bioinformatics field for years. And then this is the query I have done in January the 21st. At the time, there's really just, it's completely new, so that the closest species it can find is a bat SARS-like coronavirus, which has like 98% identity. And the second close one is the SARS coronavirus, which is about 82% of identity. But based on that, it is already very close to SARS, and it's basically we would at the time to judge that it would be a SARS-like type of outbreak. But we need more evidence to support that assumption. And also the search has time limit. So yesterday or today, I just do another blast on the same sequence, and you would see that most of the sequence are actually the variation of the current different variation of the mutated strain of the same virus that in the current outbreak. So that so you would totally see how the sequence can evolve over time and how its reference would evolve over time as well. So then we have our objectives of what data to be mined. So we first need to find all the coronavirus related data to dig on potential therapeutics for this novel virus. So that first, this company called Sally, actually, when the outbreak just happened in China in January, this company located in UK and has some collaboration with Gidi, so that I requested data from them, and then they are very generously give us a set of data on the coronavirus search, so that you can see this is how natural language processing work. That it is identify keywords of concepts that normally they have a dictionary of, such as the drug names or species name, and then they research for their logic relations based on certain wording, and then they will be able to identify therapeutics that that inhibit SARS or certain like important genes up or down regulate this virus, so that they can lock down potential therapeutic target or agent to follow on. And this is like a nice graph they generate that basically sees the associative relation between different type of reagents and genes related to SARS or other coronavirus disease, and how they correlated in the form of network. So you can totally see if we really dig in the entire database, most of the drugs are multi-target and most target are multi-effect, so that we would have a very broad and complete view of what's going on. And another similar effort is called another company called Dimension AI. They are also very key on natural language processing. They are tracking all the publication of coronavirus on the second base. Basically, this is accurate down today that the publication reached about 12,000. So that for any researchers to catch up that amount of publication would be impossible so that we need some kind of extraction. So what they did is they currently focus on clinical trials to generate a publicly visible Google Doc. And this is how the Google Doc looks like, that it's extract the clinical trial ID of all the currently, like as of today, we have 1,495 trials globally, and then they will have all the detailed information of the trial and which phase it is and which country it is. And what is the like the intervention, which means either drug or therapies or any other intervention that the trial was focused on. So that give a real-time tracking of what's going on in the research and clinical field. Another one is also very interesting, that is called the Corona Y. Corona Y basically it's a global volunteer group that initiated on this Kaggle challenge. That basically the challenge was to use NLP technology to tracking all the potential therapeutics for the novel coronavirus disease. And then this volunteer group has over 300 volunteers, and actually me and some of my team are also in it. And you can see that from this application, this is actually a Microsoft Power BI interface that you would see. They would have the statistics of each medicines has ever been mentioned in the literature that is associated with either coronavirus in general or this particular SARS-CoV-2 virus, so that you will also have a real-time track of if there's any new medicine come out, or if any of the old medicine has drawn more attention, so that it also give researcher a real-time track.